This is Hello everyone. So my name's Kropielnicki and I'm going to be presenting on cell culture, and this is for the general group meeting on March 24th. So a little bit about me. I grew up on an acreage outside of Edmonton in a small town, and my family had a lot of different animals. We had sheep, cows, horses, pigs, rabbits, chickens, cats, and a dog. So it was a super great childhood filled with many animals. And then I moved to Edmonton to pursue my Bachelor of Science degree with specialization in immunology and infection. And I'm super excited to be graduating really soon here. And yeah, here's a photo of me holding onto a sheep with my sister, and it's a little lamb, and we're feeding it. And yeah, we really love the lambs. They were all our friends, so. A little bit more of an intro. So, as well as science, I also have a huge passion in music. I really thank everyone so far who has checked out my music as well and listened to it, and all of your comments mean the world to me. So if you want to check out more of my music, I also made a little QR code here. If you want to check it out, I would really appreciate your support. So let's dive into cell culture. So the cell culture team is Dr. Upasana Singh. She is the head of the cell culture lab. She's my mentor, and she's taught me so much. And I am the research assistant, and I am doing the cell culture work for one of my Bio 499 research project courses, which has been happening since September. So what is cell culture? So ultimately, cell culture refers to laboratory methods which enable us to grow eukaryotic and prokaryotic cells in physiological conditions. And overall, what does cell culture allow us to do? So cell cultures may be used to diagnose infections, also to test drugs, and also in research as well. So it's a really great tool for researchers today. So let's dive into which cell line we are currently using in the cell culture lab that we have here. So we have HEC293 cells, and this is a human embryonic kidney cell. So these are super important because they're used in biotechnology to create therapeutic proteins, retroviral vectors, and also for chemical safety testing. We also use them for gene expression, which I'll talk a little bit about as well today. Well, a lot about. So with HEC293 cells, a little bit of an example where they're used. They're used actually in the manufacturing process for certain COVID-19 vaccines, such as the Johnson & Johnson. But it is important to note that these HEC293 cells are not contained in that final product of the vaccine because there was some controversy over that. So how and why do researchers keep using HEC293 cells for cloning and cell signaling? So HEC293 cells are used for both transient expression and stable expression, and I'll talk a little bit more about that in the coming slides. Also, these cells are super robust, and they develop every 34 to 36 hours. So they are really great for that, and they're also amenable to transfection. So allowing these to be transfected with various physical and chemical methods, which I will also talk about. So utilizing cell culture in our research. So we are expressing recombinant angiotensin converting enzyme, which is also known as ACE2. And this is found in the human body, ACE2. And why have we ultimately chosen ACE2 for this research? Why are we expressing it in these HEC293 cells? Well, ACE2 is associated with various diseases. The list goes on from diabetes, chronic kidney disease, and more. And also, it's very important for COVID research. So ACE2 has also been identified as the receptor for SARS-CoV-2 viral entry into various cells within the human body. So on the right-hand top corner, there is a picture of COVID, and they have the spike proteins there, and ACE2 essentially binds and the COVID is able to enter the cells through this ACE2 receptor. So super important with COVID research as well. So ACE2 is highly expressed in capillary-rich organs such as the lungs and kidneys, but also in the gut and the brain. So if we think about ACE2 being expressed in the kidneys, it can be detected in the urine. So from there, we think of how about making a colorimetric assay? So there hasn't been a colorimetric assay available yet for ACE2. So we're currently trying to address this issue. And by making a colorimetric assay for ACE2, we're able to detect and quantify this ACE2 in accordance with various diseases. So I wanted to extend even a bit more on this ACE2 and how important it is for COVID research. So if we can express this enzyme substantially in our lab, it could lead to drug screening and development. So we would essentially utilize the ACE2 enzyme, which we create, to test whether drugs cause upregulation or downregulation of ACE2 during treatment. And also, ACE2 is a very hefty price as well. It's $759 per 50 micrograms. So commercial ACE2 is very expensive, so that's why we want to create it in our lab. So the first objective of this whole research project was to create substantial amounts of ACE2 at a reduced competitive price for research purposes. So how did we go about this? So there's two different methods that we have done. So starting with transient cell lines. With the transient cell lines, they only take about 2 weeks of processing. And the issue with transient cell lines is the cells express the transiently transfected gene only for a finite period of time. So you only get that protein once. It's not going to continue to make the protein. So that's why we moved to stable cell lines, because that issue is solved. So the issue with stable cell lines is they take a lot of time to process, but the positives weigh out the negatives with stable cell lines because the foreign gene becomes part of the genome within those cells. So then it can be replicated, those cells can divide and continue to create the protein. There are two different pathways which the stable cell lines can go through, polyclonal or monoclonal. With polyclonal, there is an average expression level on the per cell basis of the protein of interest, but then when we go into the monoclonal cells, we actually are able to screen those clones for the highest expressing clone, and from there we get really nice high expression because we did that screening. So the HEC293 cell line. I'll start off with the transient cell line expression, which is the shorter version, where we want to make sure that everything is working, all of the transfection is working, the cells are growing, cell lysis was good, and then we confirm that we actually indeed had expression in the end. So the strategy behind the cell culture, so there's a basic overview. So we start with a cell stock or bank, and then we thaw them, culture and passage the cells, run our experiments and assay, then we again preserve those cells back into our cell stock, and the process begins again. So with the plasmid, which we are using for the HEC293 cells, we're using human ACE2 expression plasmid vector pcDNA 3.1 with a his tag as well. So this is the plasmid we're using for transfection. Here's some photos on the left of transfection. I try to take as many as I can in the cell culture while also trying to make sure that there's no contamination during that process, but I wanted to make sure I have some kind of photos to represent. So with the transfection, just a general overview of how a transfection works. So we start with the plasmid, we have a transfection reagent, we essentially insert it into the cells take up the plasmid as we incubate them at 37 degrees Celsius with 5% CO2, and then we can harvest those cells. After we harvest the cells, we want to run a Western blot to see if the protein was expressed. So we have those HEC293 cells, and then we do a protein suspension. We have the protein of interest in the supernatant. So then we run it through an SDS page, and then we run it through a Western blot through electrotransfer. Then we antibody probe it, and then we run it through the chemi imaging. And then we come to see this Western blot photo. And in this case, ACE2 was expressed, and we can see it here at 130 kDa. And this overall confirmed that the process and transfection is functional through transient expression, so we can therefore move forward with the stable cell lines, which we did. So this is the monoclonal and polyclonal stable cell lines overview. So again, very time-consuming, vigorous process, but we get a large-scale production. So this is the stable cell line workflow. So what we do is we start with a kill curve, and we determine the optimal selection antibiotic concentration, and then we move on to transfection, as I had explained earlier, and then we select a pool of transfected cells with a certain antibiotic because it's a selectable marker within that plasmid, and then we move to stable cell screening and selection. And then we do cell line characterization. So again, run those Western blots, validate that the clones are stable and very productive. And then we do expansion and downstream evaluation. And we, of course, we want to cell bank all of those clones so we can continue using them in the future. So this is for the kill curve, the first step. So we determined the minimum antibiotic, which was needed to kill all of the cells. So we have on the left a picture of cell death, or they look like they're cell rounding, as opposed to the picture that we saw earlier, the cells were nicely budding. So this shows that the cells are dying, the antibiotic is working, and we found the minimum antibiotic needed to kill those cells. But we also wanted to make sure that we have a proper graph for this. So we ran an MTT cell viability assay. So first you start with your cell seeding, you run it with the MTT solution, incubation, you run a colorimetric reaction, you add your DMSO, and then you run your absorbance reading and find the wavelength, and from there create a graph and find the minimum inhibitory concentration. From there, now that you have that, we ran the transfection and cell growth. So we have the G418 antibiotic for selection of just those transfected cells. All the rest that weren't transfected, they will be killed. So after that, this moves to the monoclonal stable cell lines. So this is where cell isolation comes into play. And we use gel-like methylcellulose to isolate those single cells. And in this photo, it's a petri dish of these cells in the methylcellulose. And we can see some single cells, and soon enough, we will pick up those cells and then we will start to screen them. So this will take a bit more time to achieve the highest expressing monoclonal colonies that we're wanting. So right now, we are on this step, the stable cell line screening and selection. For the polyclonal stable cell lines, we did finish this step. So we were able to extract the enzyme with cell lysis, and in this photo on the right, this is the cells, and they are being lysed. And then we take that enzyme and put it into our assay, which I'll talk about soon. So we were also able to cryopreserve these polyclonal cells for future uses as well. And this is an overview of cryopreservation. Ultimately, you have your media with your cells. You make sure that you're cell counting in a proper amount, and you suspend them in freezing medium. And we also use, what I love is the Mr. Frosty here. I just love that name. And we transfer them into liquid nitrogen storage. So I talked a little bit about SDS page as well as Western blot, but there's also an alternative for the identification of ACE2, and this is MS sequencing. And this is the gold standard because it really guarantees that, okay, we definitely do have expression. So this is in progress for the polyclonal stable cell line. This is an overview, a bit of how MS sequencing works. I know there's some highly professionalized people here who know a lot more about MS sequencing than I do. So I won't talk too much about that, but we will confirm our expression through MS sequencing. All right, so now that we have the enzyme, we move on to the second objective, which is creating a colorimetric assay for ACE2 quantification using this ACE2 that was created in the lab. So this is the assay method. So this is retrieved from a paper for fluorometric method. And the first step is that we have the substrate angiotensin 2 here. And ACE2, the enzyme, cleaves the phenylalanine from the substrate, and then when cleaved out of the substrate, phenylalanine is then in the matrix, and then the enzyme PAL reacts and makes a multiple enzyme reaction here, and then glutamate dehydrogenase reacts and forms NADH here, and then this, again, it makes the fluorescent product. So we can see that essentially without enzyme, it should be blue. With enzyme, it should turn a really pretty pink color. So putting papers together and figuring this out, the ACE2 colorimetric assay was created in the lab and it was very successful. So a really nice color gradient formation did come up, and we can see with no ACE2, super blue, and with ACE2 very pink. This is probably one of the most beautiful colorimetric assays I've witnessed, so it was super exciting to see this. And ultimately, the next step for optimization would be to improve the wavelength signal, so ultimately determining the best wavelength for quantification by colorimetric analysis. We also want to improve the incubation time. Currently, it's about 60 minutes to incubate to get that full color that you see, but it is a fair incubation, but we can always make this better with some modifications to the assay and also purification. So we will purify the ACE2 with the his tag, and yeah, we will have purified enzyme. And this is the ACE2 detection assay, just the standard curve seen here. I have some assorted photos as well to show you a little bit about the cell culture room. So we've got the water bath and also the biosafety cabinet with some DMEM here. This is the microscope with the Rosk flask and the cells growing, and also the incubator as well, set to 37 and 5% CO2. And here's some more sorted photos as well. So this is subculturing and passaging, which we do a lot to make sure that we don't have two confluent cells. Once we have 100% confluency, we want to make more flasks. And also cell counting and viability assay. So here's the cells under the microscope with some trypan blue to count how many cells we have. And this is also an overview. I love using BioRender to create these kinds of steps. But yes, this is the cell viability assay, just to measure the proportion of live cells versus dead cells. So the summary of ACE2 expression via the staple cell lines. So this has been a project for my research course from September to March. And what we've done was transient cell line expression of ACE2, polyclonal stable cell line expression of ACE2, the colorimetric assay for ACE2, it's almost done, and as well monoclonal stable cell line expression of ACE2. So we are going to pick up those cells and start making some monoclonal colonies. So I'd like to thank Dr. David Wishart for supervising the project, as well as his continued support in both my music and science career. It means a lot to have a supervisor who asks you what your plans are after graduating, not only with your science career, but your music career as well. So I appreciate your full support, Dr. Wishart, in all that I do. And as well, I'd like to thank Dr. Upasana Singh for being an inspiring mentor in the laboratory. I've learned so much, I've gained so much knowledge, and I can't wait to see what's to come in the near future as well. So thank you so much. And thank you